cure that we have to date to treat epilepsy. We have medications, they suppress the seizures and the symptoms, but the only cure is to find that focus and surgically remove it. So here is a photograph, not of this boy's, but a similar child or adult brain in the operating room. These are the electrodes that I talked to you about that sit on the brain surface. Each one's about 4 millimeters in diameter and has a wire coming up, and we know exactly where it is on the brain. And we record not just acutely, but sometimes days, if not a week or more, to identify and allow the patients to have the seizures like you saw in the movie, so that we can identify not only the parts we want to remove, but there's some actually parts that we want to keep there as a part of the surgical procedure. So the idea is to remove the bad parts but keep the good parts. And here's a sample electrical recording of what we see during a seizure. These are 1 second intervals. You can kind of barely see these lines here. These are these sort of spike and wave or sharp wave discharges that kind of build up in frequency and spread over time, and that's the seizure. Now, when we look at these recordings, talk about big data, there is so much data. We sample at about 1,000 hertz, so 1,000 pieces of data per information at each one of those electrode locations over about a week. It's a lot of data. And most of the time, we see what looks like white noise. You know, what is all this stuff? This is what we call interictal or between seizures recordings. And it's not normal, because in a normal brain you won't see all of these spikes that you're seeing here. So again, each of these lines represents one of those electrodes, and you can see here alignment of all of these spikes in all these different channels. And as I'll talk to you about a little bit later, in order to actually see, and this is from direct brain recordings, if you want to see a spike coming all the way through the scalp and the skin and the skull, you need about 10 square centimeters of brain that's synchronously firing just to see that potential. So one of the challenges, and what brings me into the engineering world quite a bit, is to collaborate and develop algorithms to try to turn this white noise into something that we can measure. And then when we look at our big data in terms of other things, when the tissue comes out, we can look at molecules, genes, and things and relate these together. So this was one of my MD PhD students who didn't even have an engineering background but learned how to use MATLAB and came up with an algorithm to mark the spikes, what's a spike, what's not a spike, plot them back on the three-dimensional surface of that patient's brain, because everybody has a different shape of the brain, so we need to reconstruct that from the MRI. And then we can pseudo-color areas that have a high frequency of the spiking between seizures, interictal spiking, and areas that have low in green. The circles are the electrode locations. And you can see very interesting things you can actually learn about the wiring of the brain by looking at these abnormal potentials. And you can see from some of the publications, we even publish in journals like IEEE, whatever it's called. So, so one of the questions we've been asking more recently is rather than this sort of simplistic notion that we want to just count spikes, there's a temporal aspect to it. I've been very fortunate to work with Biswajit Marathi, who's a PhD student in my lab, together with Jim Patton, and we started to ask the question, what are these spikes? Are they really asynchronous, randomly going around the brain, or are they entirely synchronous? What's going on? So Biswajit has been coming up with different ways to look at these spikes from these long-term recordings. So here's an example. You can see this is where the electrodes were placed on that patient's brain, and that surface, each one of these red areas is electrode. Then you can look at each of the channels, and it's blown up here to show that if you zoom in on some of these spikes, they're not truly synchronous. There's an actual order, A to B to C to E, or A to C to B to D, or whatever. And what he did is he used this function called a direct transfer function to look to see if there's a common pattern of propagation. In other words, is the wiring totally random, or are there certain ways the spikes propagate versus others? And using a probability matrix, using statistical significance testing, basically simplified this problem to a lot of white noise into a directional spread, and simplifying in this case for these set of 16 electrodes, this electrode number 100 tends to be where most of the spikes actually start and then spread elsewhere. So trying to take this white noise, these complex patterns, reduce them to clear numbers and temporal relationships so that we can actually then apply analytic methods. One of the challenges of big data is putting the data and integrating it together in such a form that you can actually run AI, deep learning methods on it. If you don't put it together, or if the data is not carefully curated and has lots of mistakes in it, you're going to have a lot of noise on the other end. So this has been very exciting work Biswajit has been doing. One of the things that we've learned is if you take 3 different 10-minute time periods and look at the pattern of spike propagation from this particular patient, patient 187, it's very reproducible. So one of the things about epilepsy is once you develop a focus, that wiring pattern is fairly hardwired. It's not constantly changing. If you took 3 different time points and you look at these patterns, and you can see by the shapes of the pattern, they're very consistent over time. So once you form these abnormal wiring networks, they stay that way. And here's another example from another patient, and this just statistically shows, except for one outlier, which I don't know why this one was an outlier. But anyway, we get very, very good correlations between different time periods, suggesting that once you do something to the brain that creates these epileptic circuits, they stay that way. We know as a disease, once you get epilepsy and a patient comes to me and says, how long do I have to take this terrible medication? I often have to say, for the rest of your life, unless we could do a surgical procedure like this to remove the focus and cure you of the disease. So here's just an example of a patient who has a disease called tuberous sclerosis, where they have these benign tubers in the brain. It's a genetic disorder. The little faint pink areas there are the tubers. This is showing the superposition of the photograph of where the electrodes were placed on the patient's brain. And then the size of the circles here reflect just looking at the frequency of spikes, the spikes between the seizures. Interestingly, where the seizures start is actually in a different location. But if we now apply this DDTF function on it, you simplify from spikes just occurring everywhere at certain frequencies to a directionality and in a single location in the brain where most of the spikes seem to be actually generated. So this may be very helpful in designing better surgeries, understanding the relationship of different spikes, and as we start looking at some of the other data that we're integrating and relating to this is going to help us understand more about the molecular and other pathways as well. So that's, I think, an amazing opportunity afforded to us by patients who have this disease. One of my professors, a guy named Miller Fisher, always used to call patients experiments of nature. So if somebody comes in with a stroke on the left side of the brain and we realize that that controls the right side of the body, we didn't make that happen, but we learned what the left side of the brain did. So every time, one of the things that I'm very excited about taking care of patients with many neurological disorders, I'm learning about the brain. So this is an amazing, the fact that we do surgery, that we have this degree of electrical recordings, we often get tissue at these sites, gives us an enormous opportunity that we really can't do in any other human brain disorder. So we have some of the best electrically mapped brains in the world. We have, perhaps in that building I showed you, the largest collection of each piece of brain tissue came from each electrode location, so we can map those. And we can take advantage of this now to build a way to understand the human brain and its diseases, really like we can't do. And this is what we call our systems biology of human epilepsy project. So linking many, many different systems together and creating integrated data sets that allows us to apply analytics and understand the brain. So how do we do this? So at each of these, so say this patient comes in and has surgery and we remove the temporal lobe, which is very common. There were 8 electrodes sitting on the temporal lobe, some that have a high frequency of spiking, some that have low. So we take tissue at each of those precise locations. We subdivide each block of tissue into half of it that we fix, so we can stain, do histology, look at the distribution of cells. The other half we freeze, so we can actually look at different molecular constituents such as protein, RNA, or metabolites. And that allows us to then link any of these molecular data to the histologic data, to the electrical data, to the imaging data, to the clinical data of that patient sub X. So, and if we do many of these, it creates a really exciting data set. So what are the kind of things that we'd like to learn about in these different tissues? Things at the organ level, or things that are patient-specific, such as clinical. Electrical, like where exactly was this? What were the spatial coordinates? We know different parts of the brain do different things, have different structures. We want to know where we are. What was the electrical activity? The more parameters we can come up with, whether it's the DDTF or spike frequency or seizure onset or shape or slope of spikes, as another student who's working with me is doing, we can put all that into our data repository. But then we have tissue, and on the tissue you'd like to know what's the structure of the cells. Is it normal? Is it the normal 6-layered brain, or is there something wrong with it? At each neuron or each cell in the brain, you want to look at the DNA and see if there are changes in chromatin. You want to look at the RNA, which makes proteins, and look at the proteins and their modifications and the small molecules, and then put them in a framework where you can link all of these things together and mine the data. And that's what we've been doing for 10 or 15 years now. Total data integration, the big data dream. Let me tell you a little bit about where we are so far. This is sort of the simplest structure of the database that we have currently, where we have clinical information from the epilepsy surgery program, what's the age of the patient, medications, what's the physiology at the different sites, what did you see on imaging. We need to keep track of which piece of tissue is in which box and which freezer. All of our what we call omics, genes, proteins, molecules. And the goal of putting this data together really has a practical approach, is we want to develop what's called biomarkers, things that we can use either invasively or non-invasively to characterize disease and develop treatments, and of course develop new treatments. So I'm going to talk through both the database structure and how we mine this database for the rest of this talk. So here's a sample homepage for our systems biology epilepsy project. We have a search engine. We have to make sure that all of the data is HIPAA compliant and de-identified. This is a patient. We pull up patient 150. He's a 33-year-old male. He's African American. He has a subcortical heterotopia and was right-handed. That's sort of our clinical information tab. We also have under this what medications he takes. So in terms of pharmacogenomics and things like that, we can say what medicines this patient's on versus other patients and what's going on with all these other variables. What do we find on our imaging? We have MRIs, we have PET studies, we have CAT scans. We know how they did with surgery. Did they do well? Did they do poorly? This may be helpful in guiding diagnosis in future times. And what do we know about the seizure? So these are just basic clinical variables that have to be entered into the system. We do complete battery of neuropsychological testing. So a lot of the patients have comorbid depression, anxiety, have learning problems, have memory problems. We keep track of all of these, and we have scores of all these from our neuropsychologists. That's standard part of our clinical care. We do it before and after the surgery. Anything you want to study, you can click a button here and download the data to your laptop. But most importantly, we only collect tissue and we only study patients where we have these detailed electrical recordings. If you don't have an electrode sitting on the brain and you say, well, that's from an epileptic patient, you can see by those pictures, if you're off a centimeter, it may be non-epileptic tissue from an epileptic patient. So we need to know exactly where the electrodes were and what was their firing frequency at that location. And so that's the only time we will collect tissue. And that's what makes this tissue bank so valuable. So if you click on, so we create a 3D model for every patient. You can click on a location on this program. It'll tell you which electrode it is. It'll link to the XYZ coordinates. You can go back to all the imaging studies, tell you about the slope, the shape, anything you want to know about the spikes at that location. We keep tissue inventory. So you say, well, I'm really interested in spikes that have a certain slope. Do you have any tissue I can look at that have spikes that have a really steep slope? Yes, we do, and here's what box, and we have frozen and fixed tissue. We keep 2D images. So we keep pictures with and without the electrodes in place to make sure we have the right tissue in the right place. Every block of tissue that comes out, we do histology. We stain it so we can see the organization of the cells and neurons. The thumbnails, you click on them and you download a high-resolution version. We inventory the RNA, protein subfractions, and genes. So most of the work we've done has been using microarrays that look at about 43,000 different genes, covering most of the coding human genome. And for each block of tissue, does the gene go up, does it go down, what's the false discovery rate, and so forth. And so if you want to download this data, you click Excel and you dump 43,000 genes and what happens to them in that particular patient. So this actually started at my previous job at Wayne State University. I brought it over to the University of Illinois with me. Fortunately, we just completed the renovations of the 6th floor, half the 6th floor in the Neuropsychiatric Institute. We have a place where tissue comes in, and in addition to epilepsy, we study other diseases. So we also get postmortem tissue from various diseases. It comes in, we store the tissue, we scan it digitally, we do our molecular analysis. We have histology workroom to share data and look at the histology. And then we have a bioinformatics area. And that's where we try to get people from different disciplines together, working side by side, both processing, quantifying, and mining this data. So please come by some point. And the goal is really to get the big data and to make biomarkers and therapeutics. So now we're at the big data stage. And this is kind of a different type of big data. A lot of the medical world, you talk about big data, you talk about a gene polymorphism and hundreds of thousands, if not millions of people with a disease, and you have one or two variables, you look at many, many patients. Here we look at not that many patients, maybe 10, 5, 20 patients, but thousands and thousands of variables all integrated in three-dimensional space. So the major challenge with big data that we may have heard a little bit about is how you mine it, how you find something useful, how do you get those needles in the haystack. So I'm now going to sort of shift gears and not talk about how we develop and put the data together, but what are some examples of how we've mined this data and gotten something useful out of it? So a very simple question is, why does this part of the brain produce seizures? Nearby parts of the brain don't, and here's the recording you can see. So we can look at thousands and thousands of variables in that one piece of tissue that produces seizures. What's the best control? Anytime you measure something, you have to say, what are we going to compare it to? So what would be the best control? Well, maybe a patient who doesn't have epilepsy. Well, it's unlikely we're going to be putting electrodes in their brain and taking their skull off unless there's volunteers in this room. You know? So really, and you also want to make sure that they're taking the same medications, that they have the same genetic background. We can do this in animals, but how do we do this in humans? So we think the best control is actually an internal control, the same patient's brain, where all of those variables are exactly the same genetic background, same medications, similar location in the brain, but we compare one electrode where we see seizures to one nearby that does not. And then we look at thousands of variables and ask, is there anything different between those two pieces of tissue? So that's our internal control study design, and it really works nicely because it keeps our N really low. We don't have to have 10,000 different patients to find things. And here's an example. So this is the way that we look at genomics, or one of the ways we look at genomics. They're called microarrays. It's a microscope slide that has lots of spots on it. Each spot is a different gene sequence. And when you apply the brain tissue after you process it and put a fluorescent label on it, if that gene goes up, it turns red. If that gene goes down,